Kingdom Monera. Kingdom Monera first appeared in Copeland's Fourth Kingdom classification, and later it also travelled to the Fifth Kingdom classification by R. H. Whittaker. And later this Monera Kingdom was split into two as Archaebacteria and Eubacteria in Carl Woos's final classification. In today's video, we will focus in the depths of Kingdom Monera. Without further delay, let us go into the video. If you consider Kingdom Monera, all organisms are prokaryotes. And next, all organisms are unicellular. These organisms were first observed by Anton von Leeuwenhoek. He called them animalcule. The Monera family members do not have membrane-bounded organelles. So if you name some membrane-bounded organelles, the ER, mitochondria, or the Golgi apparatus. The members of the Monera Kingdom does not have any of these membrane-bounded structures. Instead, they have 70S ribosomes. If you correctly remember, ribosomes are organelles which are not bounded by membranes. Ribosomes in the sense, ribosomes consist of ribose sugar for protein synthesis. This is very helpful for the living of a bacteria. The next characteristic is a rigid cell wall made of peptidoglycan is present. Normally in other eukaryotes, cell wall is made of cellulose, but here it is made out of peptidoglycan. Peptidoglycan in the sense, this compound is formed by the union of 4 amino acids plus sugars. Thus, a heteropolysaccharide or the word murine is given. Sometimes, other than the peptidoglycan, substance known as glycocalyx is also present. This glycocalyx is made by lipopolysaccharides. And as I said before, they have no well-defined nucleus, therefore their DNA is naked, or their DNA is not enclosed in a nuclear membrane. Members of this Monera Kingdom are almost heterotrophic, but some are autotrophic. Remember, in my previous video I said about autotrophic means there are two types of autotrophism, chemoautotrophism and photoautotrophism. And I also said only Monera members possess both abilities, chemoautotrophism and photoautotrophism. Therefore, the Kingdom Monera members are considered maximum nutritionally drivers and autotrophs in the sense they should have some pigment, right? They have a special pigment known as bacterial chlorophyll. The use of bacterial chlorophyll is that they help to trap light in bacteria. The next point is motility. Motility means locomotion. Members of the Monera Kingdom mostly locomote by using flagella. Some members even crawl or glide. And if you say examples for the Monera Kingdom, bacteria, archaebacteria, cyanobacteria, mycoplasma are some examples. If you consider the general characteristics of a Monera Kingdom based organism, they are as follows. The first characteristic is glycocalyx, which consists of polysaccharides are found. Glycocalyx is a third protection layer. It is made of polysaccharides. It protects the bacteria from drying off or phagocytosis by other bigger molecules. So consider the following diagram. And also you might consider the protection layer part I said before. So for an organism cell, the first protection layer is the cell membrane. And later they have a cell wall. In the bacteria's case, the cell wall is made of peptidoglycan. And only in some bacteria, this compound known as glycocalyx is present. Only in some, not in all. This glycocalyx acts as a third protective layer. Glycocalyx also can be two types. They are categorized by considering their appearance. Glycocalyx are found in two appearances, with a thin structure or a thick structure. If it has a thin structure, it is called a slime, while if it is thick, they are called a capsule. The next point would be the presence of a cell envelope. Normally, in eukaryotic organisms, cell envelope means the summation of the cell wall and the cell membrane. But in a bacteria with a glycocalyx, the cell wall consists of three layers: the cell wall, the cell membrane, plus the glycocalyx. But in some bacteria, the glycocalyx is not present. Therefore, cell wall and the cell membrane builds up the cell envelope. Only in some bacteria. Next is the presence of cell wall. They have a rigid cell wall made of peptidoglycan as mentioned before. The next is the presence of a cytoplasmic membrane. Cytoplasmic membrane is also known as a cell membrane. Now consider this point very carefully. Cytoplasmic membrane is made of phospholipids and protein layer, and sometimes they form mesosomes. These mesosomes are end folds formed inside the cytoplasmic membrane. Consider the following diagram. Sometimes the cytoplasmic membrane folds into enclosures like this, and then they have the cell wall and the glycocalyx. Only in some bacteria. They don't just form these enclosings. In fact, they have special functions too. These mesosomes carry out important functions such as respiration, carrying out the Krebs cycle, or helping the bacteria in cellular division or in replication. In our body, some of these functions like respiration or the Krebs cycle is carefully done by the mitochondria. Here in the Monera Kingdom, since the mitochondria is absent, the respiration and the Krebs cycle is taken place in mesosomes. The next character is the presence of a nucleoid. The nucleus of a bacteria is made of naked DNA, RNA and non-histone proteins. For a DNA to pack, histone proteins are important, but in a bacteria, non-histone proteins are present. So, the packing of DNA does not occur here. So, the number of chromosomes produced in this nucleoid is only 1. Therefore, bacterial cells are haploid. Some non-histone proteins which make up the nucleoid are polyamide. If you consider this as the nucleoid, there is another extra nucleus-like substance inside the cytoplasm. This one you call it as the extrachromosomal DNA, also known as a small or circular DNA. This small or circular DNA does not make chromosomes. They carry out special functions like giving resistance against antibiotics, and the necessary factor for that purpose, or the R factor, is present inside this extrachromosomal DNA. They also have an F factor. The F factor is the fertilizing factor. If the F factor is present, it is considered as a male, or if the F factor is not found, it is considered as a F negative or a recipient bacteria. The final characteristic is the presence of inclusions. Inclusions means food storage. In this sense, food is stored in vacuoles or metachromatic granules. The main source of food stored here is phosphorus and some sugars. Normally, if you mean a vacuole, the thing that comes into your mind is a sap vacuole. But here in Monera, the sap vacuole is not present. Instead, a gas vacuole is present. Next, let us look at the primitive structure of a Monera Kingdom member. This is the primitive structure of a Monera Kingdom organism. This part here is the capsule. And here you have the cell wall made of peptidoglycan. And here you have the cellular membrane. And here you have the nucleoid. And as you know, the nucleoid is made of DNA, RNA and non-histone proteins. And the particles in the round shape is the 70S ribosome. All these structures you have seen until now are interior structures, but there are two extra exterior structures in the Monera Kingdom organisms. One is the pilus, and the next is the flagella. Let us take a look at these exterior structures in detail. If you consider flagella, it is for motility, and a flagella has three parts, namely the hook, the filament and the basal body. The largest part in a flagella is the filament. The next exterior structure is the pili. Pili is used for conjugation. A pili is made of pillin and it is used to the attraction of F plus and F minus bacteria. Now consider a bacteria having the F factor of fertilization, and if it wants to attract another bacteria with an F minus or without the fertilization factor, they use the pili to go and attach themselves on the F negative bacteria. And next is fimbriae. Fimbriae is used for attachment. Actually, fimbriae are structures like bristles and they are much shorter than a pili, and they are used for attachment purposes. And that's it for Kingdom Monera. And if you like this video, please give it a thumbs up and subscribe to Brainiac and leave your comments below in the comment section. And also do not forget to subscribe to Funfetti Kitchen, who are our sponsors, and their link is also given in the description box. So don't forget to stay subscribed to Brainiac, because in the next video we are going to talk about more about the Flagella. Thank you for watching the video and bye. Well, my dear viewers, this week on Funfetti Kitchen we're making a simple 3-ingredient chocolate cake which you would love to make. The specialty of this recipe is that anyone can make it, and it's so simple and it's done in only 3 basic ingredients: biscuit, milk and baking powder. Looking delicious, right? If you want to watch this recipe, the link is in the description. So do not miss this yummy and delicious recipe. You'll see it now only on Funfetti Kitchen.